for carrying out assignments on excellent performance. Thank you. Okay, so I think we should begin now and others may join in the while. So I welcome you all for today's webinar on bioinformatics for precision medicine. Precision medicine is changing the way we understand and diagnose and treat major life-threatening diseases. The transformation is driven by high-throughput molecular data that is being collected from patients, animal models, and large-scale cell line experiments. In this program, we will explore how the various omics data types generated in these studies can be analyzed and integrated to study the basic biology associated with viruses and their hosts, cancer onset and development and outcomes as well. My name is Sonalika Ray, and I'm the Omics Logic Community Manager responsible for daily interaction with thousands of users that are a part of a bioinformatics community managed by Pine Biotech. Before we start with the talk, I would want to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Omics Logic is an international program that is running in 5 different regions with over 10,000 users around the world. Due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. This online program includes weekly sessions with an instructor, online tracking and review of student progress, access to all the resources and materials online for self-paced learning, access to expert guidance in problem solving, educational and research pipelines, and domain data analysis on our big data server for 15 days. Let me introduce you to the program mentors. Dr. Harpreet has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases, and looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar, who is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia, over the past several years, he has been developing collaborations with universities as a mentor and project lead for Omics Logic Training and Research Fellowship. Dr. Jepuliano Chavez, postdoctoral fellow at University of Chicago, he is skilled in bioinformatics and an expert in glioblastoma. Now, to speak about the program and why bioinformatics is a critical component of precision medicine, let me pass it on to CEO Pine Biotech, Ilya Brodsky. Over to you, Ilya.
Thank you, everyone. Thank you, Sonalika, for the introduction. And I wanted today to speak briefly about why bioinformatics for precision medicine, why it's such an important topic, and how can you get acquainted with some of the important challenges that are yet to be solved and the methods that could be used to address some of those questions. So before I begin, I wanted to mention that our company is a technology transfer company that originated at the Tower Bioinformatics Research Center. It's a research center from University of Haifa in Israel. And what we have done is we have taken the innovation that they have assembled together in an online computational platform and packaged it with additional training resources and project examples. And this has been a work of our team, so not only myself, but people that are highly experienced in these domains, including bioinformatics, training, content delivery, and things like that. And so as a result, what we've really built, I think, is a community that is beyond just the training. And the goal of Omics Logic programs is actually to provide the training in bioinformatics but it is to enable independent research that is guided by experts and peer example. And this growing community, I think, shares a passion for data-driven research and an appreciation of citizen science. And what that means really is that all of us can ask questions, all of us can have hypotheses. What I think unites us is our appreciation for data and evidence that we can find in that data. And therefore, we appreciate the skills that are needed to be able to ask those questions and get those answers correctly. And so as a result, I think that these programs are enabling thousands of students around the world, clinicians and faculty of all backgrounds, to develop novel and independent research using latest technology in life and data sciences. And so that's why we don't think that we've figured everything out. And so we continue to build our network of mentors and contributors to the program. And that's why it's important for us to maintain this growing community. And we have 2 social media groups. And maybe, Sonanika, you could paste the links to our LinkedIn group and to our Facebook group. So we have a group on LinkedIn called Omics Logic, and it's dedicated to machine learning, biomedical data mining, and high-throughput data analysis, and also a big data analysis group of over 2,000 members on Facebook. So please join us there, connect with us, and connect with our mentors. If you are a mentor or you want to be a mentor, please join and let us know. If you would like to be a contributor in some way, please also reach out to us directly. So let me start first by highlighting some of the things that probably many of us already know. For example, cancer, which is one of the leading causes of morbidity and mortality worldwide, is much more prevalent in areas of the Western world that has a growing and older population. And so if you think about that trend of the population becoming older and having additional comorbidities, it is clear that to address this effectively, the same standard measures that are working for everyone are not going to work for any particular individual in a very precise and effective way. And so it's very important for us to recognize this trend and to understand what are some of the ways that we could effectively address this growing problem that is going to require much more investment in new technologies to address these pressing needs. For example, when we think about cancer, we need to understand what are the drivers of cancer progression, what are the risks associated with developing cancer, and therefore develop screening and early diagnostic tools to develop develop an understanding of what are the markers of cancer risk and progression. And so in that approach, what we're really seeking to understand is how can we detect these molecular signatures that are very specific and unique to each individual, and be able to recognize that small piece of data, that signal, out of noisy, huge data sets that are generated every day for millions of patients around the world. And what that has to do with are new technologies that allow us to look directly into the DNA, understand the genes and RNA transcription, and look at the sequences of individual cells and tissues as this disease develops over time. And so what we're looking at is really a collection of complex data sets that include patient-derived cell lines, patient-derived xenograft models or animal models with human tumors in them. So the cell lines and the patient-derived xenograft models are some examples of how we can study the basic biology behind this disease. But also we can incorporate the clinical trials and some of the big data collected from real patients where we can find some signature information and start to ask questions. And eventually what we're seeing as a result of that is more precise and targeted therapy for many diseases, including cancer therapy. And a lot of these are actually changing our ability to treat people more effectively based on these molecular signatures, combining our understanding of drug metabolism with our understanding with genetic predisposition and with an appreciation of disease progression or the basic biology behind how this disease goes from non-malignant to more and more malignant and invasive, and eventually metastatic cancers that ultimately lead to death. And so this appreciation of data really is central to our ability to be able to provide more effective and precise treatment of patients. Now, unfortunately, what that brings us to are some big challenges with data that is very diverse, very different because it was generated by thousands of labs around the world, different hands, different methods and approaches. It was generated from many different types of people. We don't know all the information about those people. So really, it's a problem of big data. And this problem of big data is a different type of a challenge than what clinicians and biologists are used to. So biologists coming from a life science background are used to clean data, experimental data, data that had a design, data that had very specific parameters included in how the data was generated, and therefore you can test your hypothesis by looking at the signal in that data. And for clinicians, I think it's even much more challenging than that, because what they're used to be dealing with is data that is not high throughput. It is data that is obtained from lab reports. Those lab reports come with a medical history, with a medical background, and are contextualized by medical knowledge that that individual or that location really has. And so as opposed to that, when we come to this challenge of big data in the clinical application, we need to appreciate some things that are coming really from the research side, as well as understand the context for many different patients that we could potentially not ever interact with, but they're really repositories of raw data that we can mine and extract some meaningful signatures from. And potentially that could help us answer some important questions about the patients that we interact with. And so at the baseline of this, I think, is one of these technologies, and this technology is called next generation sequencing. And I think that for many of you, probably you've heard a lot about genomic data. You've probably heard about the applications of genomic medicine in a variety of different diseases. But I think what is important is for everyone to have that skill set and have that expertise in being able to look at the data, understand its quality, be able to find specific signal, and then to also lead to biological or even clinical interpretation of that signal. And that's really what this program is going to be about. So we're going to introduce you to a number of different ways to study this kind of data, analyze sample projects that we've prepared and curated for you to understand the specific questions and the applications of that data set, and then introduce you to some of the methodologies that will be useful for you to practically work with such data sets, understanding genomic variants, understanding gene expression and the variation in gene expression based on differential gene expression, looking at the annotation and interpretation of how these differentially expressed genes could be combined into specific biological pathways, and how can we really understand the biological pathways in the context of a particular disease or a biomedical question. And so one of the repositories that we will turn to is what's called the Cancer Genome Atlas, or TCGA. Now, TCGA is a very important repository of thousands of samples collected from patients that are that have many different types of cancer, so coming from different tissue and body sites. And what it provides us is some kind of an appreciation for multi-omics data and its role in understanding the disease stratification. So how is disease variable between different patients, patient stratification, how different patients are represented in this data set. They come from different ethnic backgrounds, different age groups, different sexes, genders, and how can we use this data to understand the disease in a much more precise way. So this is a really unique repository that actually anyone can gain access to and start downloading that data to be able to ask research questions finding associations between treatment, between risks, between different types of omics data. And so what we will address in this program is how to integrate such data to be able to find patterns that are relevant to different types of cancers. We'll also take a look at some of the algorithmic approaches that allow us to apply machine learning to extract signal from data sets that were generated in a high throughput way. And so one unique aspect of high throughput experiments is that the data has been generated in an automated way, and we can generate such data from cell lines based on, for example, response to treatment. So ultimately, the goal there is not to understand the molecular mechanisms behind efficacy of drug treatment, for example, but it's really to find associations to understand some signal, some kind of biomarker of efficacy. And that kind of information can then help us assign better treatments to patients, not by understanding and studying each patient individually, but actually running some algorithms that could bring out that information in front of the person who's delivering the treatment and guiding them in their decision making. So there we will take a look at how actually both of these are integrated. So we'll take a look at animal model experiments, cell line experiments, and then compare them to the variability in patients, looking at research articles that describe the data sets and trying to extract signal that is aligned between cell lines, animal models, and ultimately addresses the challenges with individual patients. Now, in addition to cancer, many other diseases are going to be on everyone's mind. Obviously, we live in an era of a pandemic, and so it's important to appreciate the role that viruses play, not only in the pandemic that we're experiencing right now, but also in many other conditions. So a lot of times we think of infectious diseases like SARS-CoV-2 as a virus that is causing a pandemic. But surprisingly, many different viruses are involved in a variety of diseases. For example, Epstein-Barr virus is involved in many cancers. Hepatitis can lead to liver cancer. So there's many diseases, including neurological diseases, that could be associated with either the direct effect of the infection or inflammation that is caused by chronic infection. And so we'll take a look at how other types of diseases could also be studied using technologies like next-generation sequencing to understand the disease itself, the immunology or the host response to that disease, and the variability in disease manifestation in many different types of patients because of what is actually going on in different tissues and cells that we look at through the lens of data. And so that's why I think that bioinformatics is really central to our ability to improve medicine and improve treatment that is more effective, less toxic, and guided by our understanding of these molecular signatures. And so to develop bioinformatics skills, you need to have some sense of the biology, computer science, and the statistical methods that are used to analyze such data. And so we hope that as a result of this program, what you will be able to gain is an understanding of a variety of bioinformatics algorithms and software, to appreciate the different databases of data that includes genomic data from cancer studies, infectious disease studies, and other diseases. You will appreciate the analytical techniques and approaches that are needed to analyze data, and you will be exposed to a variety of data science skills that, if you are interested in, you can pursue further in other types of training to develop a practical skill set that is needed for the age of big data. And so as an example, we have here a few omics data in infectious disease research that I think we'll also cover in many of the sessions that we'll discuss, that will include an overview of viral genomics and evolution, immune response biomarkers, as well as an overview of microbiome and its role in infection. Now, obviously, a lot of what I'm saying seems like too much to cover in a single program. And so that's why what our team has done is collected a number of program resources that we're going to be making available to all of the program participants to make this a more streamlined and effective use of your valuable time. So we will not only cover these live sessions like we're doing today, we're also going to provide you access to our training materials, fast big data analysis tools that you can run via a visual graphic user interface, curated project data sets, so you don't have to spend all of your time finding data, but you will be given some data sets that you can quickly go through to understand the point and the connection between the analysis and the biological relevance, as well as these interactive sessions that will be guided by mentors that I will introduce in just a couple more minutes. But before I do that, I want to introduce you. I know that some of you are probably already familiar with this, but I'm going to briefly speak about our big data analysis platform that allows anyone to quickly go through some of the important methods in bioinformatics and try them out on different types of projects. So we will, in a second, go into how to register and how to create an account. And once you do, we will take a look at this big data analysis platform so that you can understand that you can both learn and then use this platform for actual research projects that you might have. And so here you will find a variety of different types of omics data that you can analyze. You can run demo pipelines to understand the logic behind some of the different analyses that we will use. You will also see how you can share your analysis with someone else so that someone like a mentor can provide you with technical information about what went wrong in your analysis. And it also includes user-friendly tools to apply data mining and annotation of biological information so that you can get quick insights about the data sets you're analyzing. So to really see how all of this works, let's go to learn.omicslogic.com and let's start by creating an account. And that will allow us to take a look at some of the data sets and some of the example or projects that we will cover in the program. Okay, so to go there, so Narika, if you could please share the link. Okay, so when you get to this program, when you get to this portal, learnomicslogic.com, you can create a free login. This login is going to be free for anyone. You don't have to participate in the program. And in fact, if you would like to use this asynchronously, you can. Okay, when I share my screen, let me stop sharing just real quick. So when you create your account, you will gain access to both the coursework and the platform. Okay, so here you can see that you can create an account for free. Those of you that don't have an account, you can do that now. I'm just going to sign in. And once you're in, I will show you what are some of the resources that we will be using. And later on, I will pass it on to Sonanika to also look at how to register for the program itself. Okay, so please let me know once you're in. And once you have an account, you can put yes or number 1 or something like that in the chat box so that we know to proceed. Okay, so I see people signing up right now. So if you have not signed up yet, please sign up. I want to demonstrate how the program resources are going to work. Okay, thank you, David. All right, thank you, Dr. Bashir. Okay, so hopefully you've created your account now. You will find that your account comes with a personal profile. And in this profile, the first thing that you want to do is you want to finalize your name, your profile picture, give us some description about you. And this is going to become your personalized center of your learning experience. So here you will find the courses that you are taking, your progress. If you've completed some course, you will see a certificate. You will also see your certificate separately here if you would like to download them. But importantly, you're also going to have these programs here. And so once you join one of the programs, again, you will see what are the specific things that are associated with each one of our programs. So if you go to the Programs tab, here you will find all of the upcoming programs that we have. And in this particular programs on precision medicine, we have included a number of courses and projects that are going to be relevant for your ability to learn and apply bioinformatics techniques to a variety of questions and examples. So here you will find projects like the Cancer Genome Atlas, an example with liver cancer project. You will see some projects on COVID-19. You will see some technical skills that are going to be useful as you learn to integrate, visualize, and analyze your data. And you will see a number of explanations about some important machine learning techniques that we will cover in the course as well. So take a look at this, and later on, Sonaliko will explain exactly how this program is organized to continue and pass it on. The main component, I think, that differentiates this program from some of the other programs that you will find online on similar topics is that this is not just access to these online resources. This is access to training material, big data processing, which you will typically not find in anywhere else. And this is going to give you some real examples of methods to process your data. And so to briefly introduce this resource, all of you that have an account, you can use the same account to access a demo pipeline that I will briefly demonstrate to show you how easy you can go from learning about a specific method of analysis to actually using that method of analysis. And so to do that, I will go under the transcriptomics section, and I will open a number of demo projects that we will use during this program. So one of them is called the Cell Line Project. And so this is a project where they took a variety of breast cancer cell lines and performed gene expression analysis. So they generated transcriptomic data to understand what differentiates between different types of breast cancers. So here we can quickly process the same data, and as we go through the analysis, appreciate what are some of the logical steps that you need to take to go from raw next generation sequencing data, FASTQ files, all the way to getting a table of expression. So the main steps are pre-processing, mapping, and then quantification of expression levels. And what I'll do today is I'll build a very simple pipeline that in reality, if you were to try and make this pipeline on your own, might take you maybe a few hours, a few days, or even a few weeks, depending on your level of expertise. But you'll see how quick and simple this could be right here. So first of all, what we'll do is we'll clean up those raw reads, and we'll make sure that we remove the technical adapters from library preparation. We will then address the issue of PCR amplification that could bring in some technical variation that we don't want to confuse with biological signal. Then we will align these reads to the reference genome using bowtie 2. And then we will quantify the levels of expression to get a table of FPKM gene expression using our SEM. Once we've completed this pipeline, I can click on End, and I will take a look at some of the output data. So it's a real quick process to go from having that raw data to then analyzing that data, and you saw some of the variations to the pipeline that you can make as you go through, and then getting a result so that you can appreciate the inputs and the outputs very quickly. Once you get those results, importantly, we have all kinds of additional methods to mine this data, visualize this data, and even integrate multi-omics data to quickly understand the relationship between gene expression, genomic variants, maybe microRNA and long non-coding RNA, as well as some clinical information, given it is structured in the proper way. So we will go through all of these tools, but another thing that I want to mention about this program is that it is the resources and the live sessions and big data analysis tools and curated data sets. But another major component are really the experts that come to the program to provide you with mentoring. So if you're a researcher or even a student or a citizen scientist, and you want to develop a research project, one of the challenges that you're going to face is really getting the knowledge that you need quickly to select a project, identify a data set, translate your idea into a technical requirement that you can execute on. And there are many, many challenges along the way. And so instead of learning from your own mistakes and trying to do this by trial and error, it is always beneficial to turn to someone that has gone through the process that you are looking at and leverage their expertise to make your project more effective and more ambitious, perhaps, than what you would be able to achieve on your own. And so to briefly introduce our mentors, we have a collection of mentors that we've been building a network with, and we want to give them opportunities to share their expertise and their experience. And so briefly, I want to introduce Gipoliano Chavez, who is a postdoctoral researcher at University of Chicago and deals with glioblastoma. So Gipoliano, over to you.
Hello, everybody. Good morning. I apologize because I am in the lab and it's still mandatory to use a mask over here. As Elia was saying, I am a postdoctoral scholar here at the University of Chicago. I work with neuroblastoma. Actually, it's a type of cancer derived from the formation of the central nervous system. In kids, it's a pediatric cancer. I will share a couple of slides with you so that you can get a better sense of the types of answers that we can study with bioinformatics. So I chose a title for this: Towards a Better Classification of Neuroblastoma Tumors. So as Elia was saying, we can use a broad variety of data types to analyze tumors. I also have other projects where I deal with infectious diseases, but today I decided to talk to you about neuroblastoma because I think in general people are really interested in cancer. Also, this is a pediatric cancer, so I think it's really appealing to think about ways in which we can help kids grow more healthy and safer. So the problem with neuroblastoma, you can think from this perspective. So the tumor is formed basically of a heterogeneous mass of cells. And I want to highlight to you that there are two major cell types in a neuroblastoma tumor. These two major types are the adrenergic type, which seems like a neuron. So you can see these projections in the cell cytoplasm here. And that's what gives them the characteristic of a neuron. And remember that I said that neuroblastoma is derived from the development of the central nervous system. The other population that is very important for neuroblastoma development is the mesenchymal population of cells. These cells are not differentiated. So when I speak about the neuroblastoma, you can, sorry, when I speak about an adrenergic cell and that it looks like a neuron, you can think of the concept that it's a differentiated cell. So compared to the adrenergic cells, the mesenchymal cells are not differentiated. The mesenchymal cells are not tumorigenic in the sense that they do not grow as a tumor, but the problem is that these cells, they can differentiate into an adrenergic cell, and that is the problem, because although they are not tumorigenic themselves, they can differentiate and become tumorigenic. Mesenchymal cells are not sensitive to chemotherapy, so in fact, after you give chemotherapy to a child, these cells are going to survive and they're going to cause the cancer to relapse because, as I said, they're likely going to differentiate and give rise or start disease progression as an adrenergic cell. So this leads to the idea in the field that these mesenchymal cells are responsible for a process called a concept called the minimal residual disease concept, where you give chemotherapy to the child, you kill some of the adrenergic cells, but the mesenchymal cells, they are not killed by chemotherapy, they stay alive, they can differentiate into adrenergic cells, and therefore the cancer relapses or the disease progresses. As I said, adrenergic cells are tumorigenic and they are sensitive to chemotherapy. There are many ways in which we can classify these adrenergic and mesenchymal cells, or rather adrenergic and mesenchymal tumors. And these are some of the technologies that we use to classify these cells and the tumors. So this paper is from 2017 and people used RNA-seq. So you can see this is a technique called, a machine learning technique called hierarchical clustering. So you can classify the mesenchymal cells and they're different than the adrenergic cells. So you can basically separate or cluster the adrenergic and mesenchymal cells. You can also classify these cells using biochemistry experiments. This is a Western blot. It's based on the interaction between some of the proteins that are characteristic of the adrenergic and mesenchymal cells with antibodies. This is an immunological method. This is another method that is based on scoring. You basically associate a number with mesenchymal and adrenergic characteristics. So as I said, this is using RNA-seq data. There are also different type of classification where you use, other than RNA-seq, you use chromatin immunoprecipitation sequencing, and that's based on chromatin accessibility. So these enhancer regions are regulatory regions that interact with the cellular machinery that is responsible for the expression of the genes, basically the expression of the proteins. So it's really this is at the level of epigenetics, not genetics. So this is a level above genetics, because it is thought that the epigenetic level of classification happens before the actual gene expression. As it's illustrated here on the right part, these chromatin immunoprecipitation peaks, they can also be used to classify the mesenchymal cells and the adrenergic cells. One thing that is really interesting about these epigenetic methods is that they can just be used, as I showed with the RNA-seq. And this is a principal component analysis showing the separation or the clusterization of these, again, these adrenergic and mesenchymal cells. But here, not using gene expression, here it's using the super enhancer regions, the data that comes from the chromatin immunoprecipitation. So really what is established nowadays is that you can separate basically the group of kids that have neuroblastoma into two basic groups that are the high-risk group and the non-high-risk group. So as you can see in this survival curve, the group of kids that are classified as high risk, they have a very lower significant probability of survival compared to the group of kids that are classified as non-high risk. But once you classify the tumor of a child as a high-risk tumor, there's basically no way that you can use to predict the prognosis of that group, of that tumor. So in neuroblastoma, after a child is classified as having a high-risk tumor, you cannot predict how that tumor is going to act, whether it's going to progress malignantly or whether it will not. But one thing that gives us some hope is the fact that the mesenchymal cells may provide some sort of prognostic ability, because as I said, these are the cells that survive chemotherapy. So if we are able to detect these cells and the percentage of these cells that are present in the tumor, we think that we really have some sort of prognostic ability in for deciding what to do about the treatment of a child. So I'm going to finish now because this is basically the work that I'm doing right now, which is to develop these mesenchymal scoring systems to classify high-risk tumors. So we basically compare the gene expression, the epigenetic profiles of the mesenchymal and adrenergic tumors, and we then try to see if that has some prognostic value. So I'll briefly explain this to you because this is really the fundaments of the research that I'm doing. So this is I'm still working on this. This is not published yet. But what we can see, what our data suggests is that the mesenchymal scores, in fact, have a very good relationship or a very good association with low survival probability. And therefore, we think that this method can be used to quantify the genetic profiles of these kids that are on the high risk group, and therefore we can test some treatments, and we can propose that we have ways to measure and to determine whether a high risk tumor is going to have a good or bad prognosis. So I would like to end with that statement that we think that we can find ways to classify high-risk neuroblastoma tumors using these bioinformatic technologies and the combination of DNA sequencing and bioinformatics analysis. Thank you very much for the invitation.
Thank you so much, Dr. Cipollino. And now, before I pass it on to Dr. Mohit to discuss the session details, let's quickly have a poll launched to know you all better. So I've launched a poll and I would be keeping it on for a minute, and let's see what we get as answers. So my first question is, what is your designation, undergrad, grad, postgraduate, faculty, researcher, administration? And what is your research interest? Data science, bioinformatics, neuroscience, precision medicine, or infectious diseases? Thank you for your answers. I would be keeping the poll on for 30 seconds. Thank you all for answering. Dr. Mohit, over to you.
Thank you, Sonalika. Okay, thank you, Ilya and Cipollino, for this wonderful talk about your project and also about bioinformatics for precision medicine. Hello, everyone. My name is Mohit Mazumdar, and I'm one of the co-project mentor of the program. So as for today, let us go through the sessions that we are going to have, what actually Ilya discussed and what we have in the curriculum, how that aligns with the subject and the importance of what we discussed today. So let us go through the sessions as one by one. One second. Yeah. So the first session of the program will be on 6th July, and it will start with an overview of all the resources that we discussed, and also the access to the program. So for the first session, this will include program orientation, group discussion to learn from the participants of the program and about the research interest and objectives. So the orientation is also is to get you familiarized with the introduction to bioinformatics. So followed by this, the first session, which is a scheduled session which focuses on the problem of cancer. So in this session, we will also discuss about the complexity of cancer biology and the typical challenges that you see in diagnostics at organ, tissue, or cellular level. And we will also discuss about molecular factors that are involved in tumor development, that are there in for basic and both translational research in oncology. So in this study, where we have also few examples, like Ilya said to all of you, that we have already prepared a few examples. So in one of the examples is a study where you can see on the screen a patient derived cell line. And this in this the xenograft models are also used in another study. And we have also data sets where we use clinical trials information that offers a collection of data to understand and get information about the diagnostics and also treatment of cancer in patients. So the next session after that, which would be the second session. In the second session, we will talk about the technology after getting an overview of cancer and how deadly it is and what are the different characteristics to understand a cancer cell. So in the second session, it will be more about the technology, as I'm saying. So it would be about understanding how next generation sequencing works. You will learn about shotgun sequencing reads, and then you'll learn about how to make the data structured, and then you will learn about different bioinformatics methods that uses like processing, where we will be doing both germline and somatic mutations. We'll look at the analysis, where we will do mapping, detection, variants you can see on the screen. And also in this in the last part, so in the last part of the session, we will discuss about the interpretation. So how can you interpret this data? And along with this, like you will have access to the resources that explains all of this, so that you kind of get familiarized with what is being covered and have a chance to do it at at your own pace. So anytime that you feel that you have time to go through the resources, these are online courses that are available on the Learn Omics Logic portal. So in here, I mean, we have different resources that it starts from a very basic introduction, and then goes into different processing pipelines and how data can be interpreted and how you can structure that data. That could make sense because it's from large number of it's a big data, and then you're transforming into a very meaningful data set. So in this study, in this session, I mean, we will learn about how genomics is used in biomedical drug discovery, and how genomics is used in different other areas in translational research. So in the third session, which will be on processing of another type of sequencing data, which is the transcriptomics data. So in this session, after learning about the genomic processing data and its application in biomedical research and precision medicine, you will learn how we can measure functional proteins and look at the mRNA abundance and accurately understand the alternative splicing. So you saw examples of RNA sequencing before. So going through this entire process of RNA sequencing, doing the read quality and pre-processing yourself, going and then mapping it to the reference genome, and then understanding its role by quantifying the genes and also the transcripts. And in the same way, after doing that, you would want to do the visualization. So I think this entire process is also to let the scientific community know of what you have what you have obtained as a result. So expressing that in a visual form and getting the visualization is a very important part of our scientific things that we do every day. So in that sense, we will learn about how you can correctly and visualize the gene expression information, and also do PCA to do that, and also go through annotation and interpretation. So in this session, we will be going through the for example, the processing pipelines. We will prepare a full pipeline and then go through different type of analysis that we do for gene expression data. So the session after that, which will be the fourth session, is on machine learning. So you have already learned about overview of two different technologies, which is genomics and transcriptomics. And then you would want to learn about how do you, do this, make sense of this data and do this to find out different patterns that are lying within a large number of other data sets also. So finding predictive modeling, or like understanding how this what this data have information. So this complex collection, where this is about the data and their complex collection, we are looking at different types of characteristics. We are looking between correlation between clinical manifestation of disease, molecular patterns, and how we can define it using different statistical analysis, right? So machine learning approach allows computational power that could be applied to these challenges. So in this session, we will use machine learning. And during this time, as I was saying, that there will be resources allocated that will help you go through and do this on your own. So you will also have access to the coding platform, coding playground, which is there in Learn, where you can write and do scripting in Python and R. And these also they are also like tutorials that takes you through this entire process. So you would know how to apply R and Python in your genomic or transcriptomics data analysis. So yeah, that would be an experience, and that those will be available to you as the session goes on, and we will cover many of those aspects in these live sessions. So after going through and learning about how do you visualize and how can you actually analyze from different project examples that we have prepared for you, the next thing is to go and do it yourself, like finding those important repositories on, and finding the data sets from those important repositories like SRA, Geo, NCBI, that are there in NCBI. And you already Ilya already discussed about the TCGA repository. So this session will be about those different repositories that are there and very important for any bioinformatician to explore and understand how to extract the information that you need for your project. So we will be discussing in that respect. And about the opportunity, many of us here do not even understand the opportunity that is related to training like this. I mean, why we are doing this, why we are saying that independent research is now very much possible and within reach of everyone. Why is so? It's because the availability of data. And yes, I mean, that kind of data is available. So and NCBI is such a good example of that. You can see that how many data sets have been already deposited, and on the way it is growing rapidly every day. So after learning about the data sets and how to figure out and find data, we will gain, after the first phase, I think it would be 5 sessions. And then after the 5th session, the 6th session would be about next generation sequencing, where we will learn about viral genomes in the host transcriptome. So here we will also learn about oncolytic viruses. So that is one of the um, feature of learning through precision medicine program, is to learn about different aspects of transmissions from cancer, and then these different viruses. So here we will also learn about like how they are involved in tumor development. How do you connect from virology to cancer? And then learn about inflammation and tissue damage. Here we will also learn about how can we align, annotate, and do uh, you know mapping. So we will learn about viral genomes, and we will also learn about alignment to databases of those viral genomes. So after learning about that, so the second phase in the where we are going to learn about different aspects of how can we interpret virological data for virology and infectious diseases. So in this session which is session 7th, we will learn about multiple sequence alignment and phylogeny. So here we will learn about how can you study viral genomes for liver, blood, and neural tissues. And we will also learn to prepare data and setting up bioinformatics pipelines, where you will find to do phylogenetic analysis and multiple sequence alignment of these genomes. So I think most of you are familiar with phylogenetic tree. So phylogenetics is a study. Phylogenetics, in the study of phylogenetics, you learn about the lineages between different species and how you can interpret that. So after that, so which is the 7th session, so this is spanned across 2 sessions a week. So I think it would be all right, and we'll be going through this and you'll get enough time to complete the assignments and also go through some of the courses and materials and data sets that we have prepared for you. So learning about that, then practicing. So it's a very important aspect of this program is to do the hands-on tutorials and do the hands-on practice yourself to gain the most of it and become an expert, right? So to do that, you have to be more comfortable by on the platform that you will have access to during the 1 month program. So here we will learn about workflows and pipelines in bioinformatics. So we have discussed already, I've seen a lot of pipelines and workflows there, like algorithms that are put together in a logical manner, right? So and that in that way, we have technology that helps us to find out and detect those challenges and find out critical important information to design to design a research correctly. So this is a very important role that plays in bioinformatics, and here we will learn about detecting and mapping those to databases, and we will also learn about how you can understand about how genomics and genomes and the evolutionary time correlates. So this is something that we have planned for this session, and here we will also go through some of the processes in this. This is another one of the example. This is a birth death example of evolutionary study. So here you will also, there are like parameters that we will cover that are there to understand this sort of algorithm, and what are different algorithms to do this and how they can be applied for a problem. So about the problem, so now the next session is about the problem from infection to pandemic, from a viral adaptation. So here we will have an example about the coronavirus pandemic, and we will do take out sequences from NCBI virus, and we will do comparative studies where we will compare these 4 sequences, genomic sequences, full sequences, and find out like how they can correlate and how they make sense in activation of immune response, and then cytokine storm, which is caused by viral replication, and also could relate to what is happening currently everywhere. So as I said, that you would be able to do it alongside with us. So we will use the t-bioinfo platform, and where we have sections on to do this, which is virus quasi species. So we'll discuss this, and we'll tell you like how this is done in session 9. So in after session 9, the last session, where this entire story comes to an conclusion, where we are going to learn about host pathogen interaction. So after applying and learning about all of these technologies and how they can be done for different diseases, sorry, here we are going to learn about what are the strategies that we can apply to overcome. So we will talk about vaccine design, so how bioinformatics, or how you can consider data and then think about vaccine design. So you have a lot of information coming from big data, about the sequences, about the gene expression. Now you have figured out that this is a protein of my interest, and I think these are the possible sites, and based on the mutations that I've seen in the genomics, these are some of the places that we should target, and that these are some of the epitope binding sites. So based on all that information, the important thing is to go for data-driven hypothesis, and that data-driven hypothesis is something that we are going to discuss throughout the program. So in the session, we will cover about some of the structures. How do you connect from uh, host response to viral replication, and then implications of that to cell, organ, and tissue. So exploring different viral strategies for exploitation of host machinery and the viral protein production. So these are some of the topics and sessions that are there for precision medicine. And I think one of the best part of this is that we also will be doing giving and going through the courses, the coursework, which actually is pretty amazing, and that's what we have heard from all of our participants so far. So after doing this, and if you have any confusion, and if you think that bioinformatics is difficult, because we are integrating so many things, we are integrating data, we are integrating algorithms, we are integrating complex machine learning methods, it sometimes gets like really thought it to be like, sometimes it gets really challenging. But we are here to help you out with that, because we have gone through this entire process over and over again, and thought of different strategies that actually works. So with that, I would now maybe pass it back to Sonalika, and we'll be happy to take any question and answer. And if you have any questions, and maybe I can also look into the Zoom. So if
Yes,
there is
Dr.
any.
Mohit, we have a question. Do I need to have a specific background to take the course?
Yeah, I think there is the programs, the way we have planned them starts from the basic. So as I told you, that if you have a basic background in biology, and if you have interest, if you have keen interest in doing this, and if you have something to do with research, so I think this is a program for you, because what we will be going through is research based training. And this will take you to what is happening, not just like what we have put together, but taking real examples, like the articles that are published now. So it's actually correlates a lot with what is happening in the industry, but also what is happening in current research in academia as well. So that's the opportunity that you have. And if you would want to know and learn from the very basic, we also have the coursework to help you with that. And along with that, we have mentorship, and then we have also a research fellowship program. So it's just not that you can do everything in all of this in 1 month. I think that's quite a challenging thing to do, but it's a very good thing to start from somewhere. And this is that sort of opportunity that takes you to all of the great topics that are there for discussion in scientific community today. And then choose and understand and decide that what is that you would want to contribute to. So these are some of the thoughts that I'll leave you with. So now, Snalika, if anyone has any other question, I will be happy to answer that as well.
Thank you so much, Dr. Mohit. We have a question for Dr. Giuliano. Dr. Bashir asks, is there any mechanism to prevent the differentiation of MES to ADRN?
Yeah, there are.
Actually, he answered me the private message. Thank you for that.
Okay. Thank you so much. Okay, so if there are any more questions, you can post it on this chat box. I'm just sharing my screen. Let me just quickly take you through the pre-registration and the registration for the program. So by the email, you would have got this pre-registration link. And if you don't have one, I would be sharing with you on the chat box itself. You can pre-register yourself for the program here. Now, when I say pre-register, it's just a formality to be a part of the program so that we get your details. And on this pre-registration page, you would be able to find all the details which are associated with the program, what all would be covered, the session details are mentioned as well. And also we have answered few of the frequently asked questions. So you can just go to the pre-registration page and find your answers. Also, all the session details which just Dr. Mohit explained are mentioned on the pre-registration page. Now, as I told, pre-registration is just to fill out the form so that we get your entry. But to proceed ahead with your registration and your enrollment in the program, you need to proceed with the registration program page. Here is the link. Once you proceed with this program page, you would have to click on Start Your Application, and you would get 3 levels: beginner, intermediate, and advanced. The 3 levels, they are basically based on the basis of the provisions that you would be getting for the program. And the provisions are mentioned here very clearly. You can go through each of them and pick the level which suits your requirements the best. And we also have opportunities for people who are looking for scholarships for their research work. And those who are interested to get scholarships, they can mail me at marketing@pine.bio, and I would be glad to help you out with the scholarship process for your research work. And you can also mail Dr. Mohit at mohit@pine.bio, and we would be happy to clear all your queries. Uh, we have a question. Dipta Shri Mandela asks how frequently these programs are offered. So Dipta Shri, if I'm pronouncing your name right, we have these programs ongoing every month, but yes, different topics. So we had bioinformatics for infectious diseases, then we had machine learning, that's the ongoing one. We have chem informatics program as well, which is ongoing. But yes, we won't be repeating them. So we for the precision medicine one, it would be starting on July 6th, and it would end on 5th of August. And I hope that answers your question. And Kwok Hang Lee asks, I just began with bioinformatics. I noticed that many bioinformaticians use Mac OS. Oh, okay. So first of all, that's not an issue at all. I am actually using Windows right now. So what are the advantages of Mac OS compared to Linux or Windows? Dr. Mohit, would you like to answer this in detail, please?
I mean, I don't think that there's any
Yes.
advantages per se to Mac OS. I just think that because it's based on Unix, you can run some Linux right there from the terminal, and a lot of people end up using it. So I think that's the main reason. I don't think that you should be thinking that, you know, if you want to go into bioinformatics, you have to use a Mac OS computer. It's just something of preference.
Yes, exactly. I believe the same. So it doesn't matter whatever system you have and whatever specification you have. While talking about specification, it just reminded me that many of the researchers, they are the basic shortcoming in their research work is their specification. There are many people who are not able to actually update their systems and get the required specification to complete their research work, and that's where our server t-bioinfo server comes into play and helps out carry out different sorts of big data analysis all at once, and not giving any a thing to your system specification. Yes. Okay, so I have explained all the registration and the pre-registration forms and also the sessions. I hope are well clear to you. If anyhow you want to go through the syllabus once again in a detailed manner, we also have the syllabus mentioned on the syllabus page. I am sharing the link with you as well. And let us know if you have any doubts. Thank you so much for joining us with the session today. It was really great to have you all with us. Thank you, everyone. Bye-bye. Hope to see you soon in the sessions.
Thank you so much. Thank you.
Thank you so much, Dr. Chipaliano.
Thank you so much. Have a great day.